But right now, Dr. Lisa Crockett and the South Seas. Everybody turn off your phones. Engage in this, not social media, go somewhere else, okay? See you in the
Thank you, Sarah. Thanks, everybody, for coming. And I tried to set the stage by showing that video. Most of you were here to see the rough weather and rough seas that we experience crossing the Drake Passage on our way to work in the Southern Ocean and on the Antarctic Peninsula. And what I'm going to do today is I'm going to share with you some secrets. I call them secrets because I think that many of you are not aware of many of the things I'm going to talk about. I'm going to talk about some of the inhabitants of the Southern Ocean. I'm also going to talk about the perils that these animals, these organisms face as a consequence of human-driven climate change and fisheries that are commercial fisheries that are taking place in the Southern Ocean. I have to help me today. I have two of my graduate students, Amanda Biederman, who has been at OU. This is her fourth year at OU. She's been to the Antarctic twice. She just came back in August for 4 months on the ice. And Elizabeth Evans, who's a first-year graduate student. She was an undergraduate at OU. And Elizabeth has also been to the Antarctic twice. So we're all part of a much larger collaborative research project that we're going to talk about a little bit later today. But first I'm going to talk about, as I said, the secrets, a little bit about some of the inhabitants of the Southern Ocean, as well as the adaptations and challenges that these organisms face living in a very cold and forbidding environment. So that's kind of the nature of where we're going. So I think many of us, when we think about the Antarctic, we think about penguin, we think about the march of the penguin, and these are magnificent creatures, no doubt, but I think that many of us are less aware of some of the other inhabitants of the Southern Ocean, including these kelps. So there are extensive macroalgae forests in the regions of the Antarctic Peninsula. There are a wide variety of invertebrate animals, animals that lack a backbone, such as these cup corals, seagrasses, giant isopods shown here, and the students will pass around examples of some of these animals that we brought in from the lab. Giant sea spiders that are characteristic of the deep sea, but also live in the Southern Ocean, and on and on. And among the fishes are a group of fishes known collectively as the eelpouts. And these animals are quite striking in that I think when I see one that they'd make a great star of a children's book. They're really a little bit comical looking and sweet animals. Now again, I think when we think about the Antarctic, what first comes to mind are the charismatic megafauna. We think of whales, we think of seals, we think of penguins. And this is a humpback whale that was photographed last austral spring, just a few months ago, by a whale researcher. And what I want you to do in the next series of photographs is think about how many primary and secondary colors you see in the photograph. So I'm going to scroll through these fairly quickly. So think about how many primary or secondary colors you see here, how many primary or secondary colors you see in this photograph. These are blue-eyed shags or cormorants. These are not penguins. This is a snowy sheathbill. How many primary, secondary colors do you see here? This is a giant petrel. Again, how many primary or secondary colors do you see here? And this is a leopard seal. Again, these are, among these charismatic megafauna. So how many primary and secondary colors did people see in these photographs? Oh yeah, white. What else? Between like 1 and 3 colors. 1 and 3 colors. Who else? 1 and 3 colors. Lots of blue, lots of blue, lots of white, lots of black. Not a lot of variety. All right. Now we're going to go under the surface of the Southern Ocean. And I'm going to ask you the same question. How many primary or secondary colors do you see? This is a green sponge, a yellow sponge. I shouldn't give it away, right? I shouldn't tell you what colors they are. All right, coral and algae, encrusting sponges. How many colors are you seeing? How many colors do you see here? This is an example of some of the invertebrates that we collected in our trawl when we were fishing. So these are animals that live on the bottom, on the seafloor, and they represent a lot of very different groups of animals, invertebrates. This is a Gorgonian coral. We've got our sponges, asteroid stars, a pycnogonid, which is a sea spider. I'll tell you how to spell that if you want to know. A cup coral, a jellyfish, and so forth. So we've really got a lot of diversity, but we also have a lot of color in the underwater environment. Now, this has actually been described by a woman named Skye Murray, who was a marine technician aboard one of the research vessels. And as her master's in fine arts at Northeastern University, she actually analyzed a series of photographs above the ocean surface and beneath, and then set up these, she analyzed the pixels, and then set up these color bars, each representing an individual photograph. So this is one photograph, this is one photograph. All of these bars that are on top are from above the sea surface or at the sea surface, and all of the bars beneath are below the sea surface. And you can really see that the color patterns are so much more varied below the sea surface than above the sea surface. Now, to introduce the fishes, which are the subject of our research, I'm going to talk about specifically the notothenioid fishes that dominate not just the biomass, but represent about 50% of the species that are found on the continental shelf of Antarctica. So these are really the key players, the key figures in our research. So again, they come in all colors and stripes and varieties. This is one of my favorites, Harpagifer antarcticus. This is a very beautiful sculpin-like fish that has different color morphs. Some of them are blue, some of them are red, some of them are brown. We've got our dragonfishes. These are two different species, one species here and one species here. This species is known as the naked dragonfish because it lacks scales. And you can kind of see why it might be called a dragonfish given its long snout. We have another family of fishes that live in the Antarctic. There are 16 species of this family. Yeah?
Seems like a lot of the fish have like slimmer bodies in this. Is there a reason that was that?
That's a good question. They do have, some of them do have slimmer bodies. Some of them are much more not so slim. So the ones in the picture I'm showing you, they've been on the weight loss plan. So some of them, some of them that are a little more sluggish tend to be more rotund, but these particular individuals are fairly slim, and that reflects somewhat their lifestyle. Some of them are pelagic. Many of them live on the bottom, but some of them are pelagic, and they're swimming in the water column, and having a more slender body is energetically less costly for swimming. So this is an animal, actually, that does look fairly slim in this picture, but this is a benthic species. This is a family of fishes that's unique in the world because all members of this family lack hemoglobin. Hemoglobin is, remember, what makes blood red. It's the oxygen transport protein that, again, makes blood red. We're going to talk about some of the consequences of the lack of hemoglobin momentarily. But these are really quite unique fishes. They have white blood. They don't have red blood like all other vertebrates on the planet. So what I'm going to talk about more specifically today is I'm going to talk about, actually my students are going to talk first and give you a short introduction to the oceanography of the Southern Ocean. Then we'll talk about some of the challenges and adaptations that present themselves to the organisms that live among the ecosystems within the Southern Ocean. So these are some of the secrets that I'm going to share with you. I'm going to talk about some of the anthropogenic challenges that these organisms face, specifically as a result of climate change and commercial fishing. And finally, we will end by talking about our current research and sort of the directions we're going with our research. So to get started, we're going to have the oceanography lesson, and that's going to be done by both Elizabeth Evans. This is Elizabeth
Hi. So the first thing you might notice is I have a prop here, a globe, and it is turned right side up, so to speak, so that Antarctica is, the South Pole is facing up, just for reference. The Southern Ocean is the fourth largest ocean on the planet, making about, containing 15% of the planet's water. And something that's really striking about it is its depth. It's very, very deep. The continental shelf of Antarctica is relatively narrow and very deep, and the surrounding current, which Amanda will talk about, is also very deep. So this is important because, as Lisa mentioned, the fish that we study are benthic organisms. They live on the bottom. So the depth of the ocean floor is really relevant and important to our research. And here in the Southern Ocean, the average depth is about 7,000 meters, so pretty deep. And the only really shallow habitable parts of the ocean by these fish are very close to the continental shelf. So the Western Peninsula, which is here, so the fish are isolated to the Western Peninsula and over on the eastern coast here. Those depths are relatively shallow, about 500 meters in depth, which is really the only habitable location for these fish. And furthermore, the continental shelf, the slope is very steep. So it slopes down very quickly and very deeply, just a short distance away from the shelf, really limiting the depth that these fish and the locations where these fish can live. Okay.
And this is Amanda Biederman, another graduate student.
Hi. So as Elizabeth mentioned, the Antarctic Circumpolar Current, or the ACC, is a very important feature of the Southern Ocean. It's a large but slow-moving mass of water that surrounds the entire continent in a clockwise motion. It extends all the way down to the seafloor. And because it extends down so far, it restricts migration to and from this region. And as a result, the Southern Ocean is a very unique and very isolated ecosystem. And so this plays into the physiology and the ecology of the animals that we study.
Okay, so the first challenge that I want to talk about is what keeps these animals from freezing. And what I'm going to ask you are some questions that I think hopefully are obvious, but may not be obvious to all. And the first question that I'm going to ask is, what temperature does fresh water freeze at? Who can answer that? 0 Celsius, thank you. What temperature does seawater freeze at? Approximately. No. So if you put salt on ice, what happens? It melts. So seawater freezes at a lower temperature than fresh water. It actually freezes at about a temperature of minus 1.8 degrees C, which is 29 or so degrees Fahrenheit. Now, the reason that it freezes is because it has a very high salt content, and we can measure the salt content in a term that we like to use of osmolarity. milliosmolar specifically. So seawater freezes at, or seawater, I'm sorry, has a salt content of 1,000 milliosmolar. Now, there are two types of cold-blooded animals that live in the Antarctic, and I'm going to refer to these animals as ectotherms. So in science, particularly in physiology, we like to refer to cold-blooded animals as ectotherms because they're deriving their body temperatures from their environment. They're the same temperature, or they have the same body temperature as the outside or their environment. So a sea star is an example of an ectotherm. It lives at the same temperature. Its body is the same temperature as of that of seawater. So its body temperature is minus 1.8 degrees centigrade. It has the same salt content as seawater. So if the seawater that it's living in isn't frozen, the starfish or the sea star does not need to worry about freezing. Now, in contrast to that, most fishes don't have the same osmolarity. They don't have the same salt content as the sea stars or as seawater. So again, they're going to live at the same temperature as the seawater, so minus 1.8 degrees C, the freezing point of seawater, but their osmolarity is only about half that of seawater. So that will depress the freezing point to about 0.7 degrees centigrade, so well above the freezing point of seawater. So something has to contribute to further reduce the freezing point of the body fluids of these animals. Otherwise, these animals would be frozen solid. They'd be popsicles. So antifreeze glycoproteins were discovered back in the 1960s by a relatively young graduate student from Stanford University. And since then, they've been characterized. They're now presented as examples of biochemical adaptation in textbooks. And in the absence of glycoprotein antifreezes, a fish would freeze at a temperature of about minus 0.7, again. Not sufficient to get that animal to survive in freezing seawater. In the presence of the glycoprotein antifreezes, the fish now can withstand temperatures well below the freezing point of seawater before they would freeze. Now I'm going to show a short video that provides both some nice underwater footage, so you'll see some of the animals that I'm discussing in sort of moving images, as well as discuss some of the more current research on the glycoprotein antifreezes.
Every creature that lives in and around Antarctica has to deal with one overwhelming reality: the cold.
There we go.
Some use insulating fat and water-shedding feathers. But for ice fish living in below freezing seawater...
So this is ice, anchor ice living on the bottom, or not living, but on the bottom.
Including in the bodies of the fish. But the fish have proteins that latch onto the ice crystals and keep them from growing and destroying living tissue. And scientists have recently found that these antifreeze proteins also keep the ice crystals from melting. To figure this out, the researchers dove beneath the ice and collected fish. They drew antifreeze proteins from their blood to test in the lab. They found that even at temperatures that would melt normal ice crystals, the ones in the fish don't melt. So ice crystals must be accumulating in the fish throughout their lives, although scientists don't know exactly what the negative effects might be. Internal antifreeze is obviously a lot better than freezing to death, but it is one of those evolutionary solutions that probably comes with a hidden cost.
So that, again, I think presents some of the challenges that, that these animals face. So virtually all of the fishes, all of those notothenioi fishes, have antifreezes. The amount of antifreeze they have depends on where in the water column they're found. The ones that are living much deeper don't need as much antifreeze because of the high hydrostatic pressures. It prevents ice crystals from forming. But otherwise, all of these fishes have these antifreeze proteins. Now, the second challenge that these animals face is how they stay active. So I think when many of us think about cold-blooded animals and we think about cold-blooded animals in Ohio, we know that many animals more locally go into some sort of dormancy or period of rest in the wintertime, a type of ectothermic hibernation, so to speak. So the challenge for these Antarctic animals is that they don't have the luxury of waiting for the summer when they can start moving around and carry out their life cycle. They need to be active year-round, and many of them are active year-round. And it was noted back in 1905 by the Discovery expedition led by Captain Scott that these Antarctic fishes were quite active. So he wrote in his diary, besides what we may call the Antarctic whiting, our people caught a quantity of a surface fish that frequented the pools and cracks in the ice during the summer. I heard that it gave very great sport. So these fish are remarkably active for living at such extremely cold body temperatures. And I'm going to show again a video that was taken by one of the station members a few years back that I think illustrates that these animals are not just sitting on the bottom doing nothing. They are really moving around even though they're living at such cold temperatures. So these are the ice fish, these fish that lack hemoglobin. And when they open their mouth, you can see it's all white in the inside. So when I was a master's student at the University of Maine, so when I was doing some of my graduate work, one of the questions that I was interested in was, how do these animals stay active even though their bodies are so cold? And essentially what I did was I compared Antarctic fishes with some similar temperate fishes, similar in their ecotype, so similar activity patterns and so forth. And what I did was I wanted to quantify how much ATP could be made in a given period of time by the Antarctic fishes and by the temperate fishes. Remember that ATP is the energy currency of the cell. All of our cells need ATP to do work. We need ATP for locomotion. We need ATP to pump ions and so forth. And I compared the capacities of the Antarctic and the temperate fishes in making ATP. And what I found was that the Antarctic fishes, by far, can make ATP at a much faster rate than can their temperate counterparts. And this is in part an explanation as to why these animals can stay active even at very, very cold temperatures. They can make ATP quickly. They can then provide the ATP necessary for muscle contraction, which propels the animal through the water column. Now, the third challenge for Antarctic fishes is the story about oxygen. I'm going to tell you a story about the physical properties of oxygen. So this is a very simple graph that illustrates what happens to dissolved oxygen if we raise the temperature. So if we raise the temperature, the amount of oxygen that's physically in solution decreases. And I think a lot of us know this firsthand from if you take a nice cold glass of water to your bedside table before you go to bed, you wake up the next morning, the glass of water is warm and it's full of oxygen bubbles or air bubbles. And this is because as the water in that glass is warmed up, the oxygen has come out of solution. So oxygen is far more soluble in cold water than it is in warm water. So this is important when we think about these ice fishes, these fishes that lack hemoglobin, those 16 species of the family Canichthyeidae. So these animals were first described, at least their blood was first described in the scientific literature almost more than 60 years ago now, by a Norwegian named Johan Ruud. And Johan described these animals and showed the remarkable whiteness of their blood. And he quantified, he looked for red blood cells, he couldn't find any red blood cells, he couldn't find any hemoglobin. And these fish were originally thought to be bloodless. So back in the 20s, and these fish were just simply thought to not have any blood at all. And Ruud wrote in his paper in Nature, he wrote that only in a place like the Antarctic, I'm paraphrasing here, only in the cold water of the polar regions could a fish survive in the absence without hemoglobin. And this goes back to this relationship between oxygen solubility and temperature. So remember that the Antarctic is very cold, that Southern Ocean is very cold. There's going to be a lot of oxygen simply just dissolved in the blood plasma of these animals. And that's how they can transport and deliver sufficient oxygen to their tissues, because they're living at such cold temperatures. Well, how do we quantify how much oxygen carrying capacity, how much oxygen can be actually transported in the blood? We can do that by measuring what's called the blood hematocrit. So the hematocrit is quantified by taking a sample of blood, loading that blood into a thin capillary tube, spinning it in a centrifuge. And when you do that, the red blood cells sediment or pack at the bottom of the tube. And above that, you have the blood plasma that lacks any of the red blood cells or white blood cells for that matter. And then you express that as a percent, the red blood cell content as a percent of the total volume in that capillary tube. Well, Antarctic fishes have depending on the species, have different hematocrit levels. Some have low hematocrits, so this would be relatively little red blood cell volume compared with the blood plasma above it. This species would have a higher hematocrit, more red blood cells, compared to its total blood volume. So this is all plasma above that. The ice fishes, of course, have zero hematocrit because they have no red blood cells. So if these animals have no red blood cells and no hemoglobin, what this means for these animals is that they can only transport about 10% of the oxygen that their red-blooded relatives can. And so this presents, again, a very formidable challenge for these animals. How do they get sufficient oxygen to their tissues if they can only carry 10% of the oxygen that their relatives can? Well, there are several different strategies that these animals have evolved to cope and circumvent the problem of not getting sufficient oxygen to the blood. First, what they do is they have a greater blood volume. So if a milliliter of blood can only hold so much oxygen, let's just have, instead of 10 milliliters of blood, let's have 40 milliliters of blood. That way, these animals can transport more oxygen. Well, if you've got a larger blood volume, you need a bigger pump to pump that blood. A bigger pump is reflected in the much larger heart. So this is the heart from an ice fish that has the same body size as this red-blooded species. The heart is about 4 times as large. If you've got more blood, you need a bigger pump. You need a bigger heart. The other thing these animals have done is to reduce the distance over which oxygen has to diffuse. Now, I've put this Fick equation, those of you who've had any physiology know the Fick equation well. Hopefully you remember it. So the Fick equation is an equation that describes oxygen flux in this case. How rapidly, what is the rate of oxygen diffusion? How rapidly does it move from here to here? So this, what I've got in red here, this delta X is the diffusion distance, the distance over which that oxygen has to diffuse. By making that value small, the delta X, you're going to increase the flux or increase the rate of diffusion of oxygen. J will be larger if delta X is small. So let's look at how these animals do this. So here's what we're looking at is we're looking at the vasculature or the blood vessels in the retina of a red-blooded Antarctic nototheniioid. And you can see the patterns of the capillaries and so forth shown in yellow there. This is a dragonfish, another nototheniioid, but this animal has a smaller hematocrit. It doesn't have as many red blood cells, so it can't transport as much oxygen as could this fish. Now look what happens when I show you the ice fish that has no hemoglobin, 0 hematocrit. Wow. Look at the capillary density. It's just remarkable how this increase in capillary density now shortens that diffusion distance and enables these animals to get sufficient oxygen to those retinal cells, which are very greedy for oxygen. They have a very high oxygen demand. So this is one way that they've adapted and coped with the problem of smaller, lower oxygen carrying capacities. Okay, so those are some of the challenges of living in cold seawater. What about some of these anthropogenic challenges? So I want to talk about, this, it's not a hoax. It's not a hoax. This is a photograph that I took off the web, a NASA photograph that shows the crack in the Larsen C ice shelf. There are various ice shelves in the Antarctic. You can come up and look at some of them on this globe later if you want. But these ice shelves form buttresses for the ice sheets that are on the continent itself. You lose the ice shelves, you lose the ice sheets. It's essentially like taking a cork out of a bottle. The ice sheets will just flow, and they'll be gone, and sea levels will rise, etc. This Larsen C ice shelf broke off in mid-July. I think it was July 12th. And you may have heard about it. It was the size of Rhode Island or Delaware. Huge, huge chunk of ice. A few years prior to that, the Larsen B ice shelf broke off. The Wilkinson Wordie ice shelf broke a few years before that. They're breaking all the time. And this is not a Chinese hoax. In our own backyard at Palmer Station, this is the station. This image shows the retreat of the glacier, again, right in our very backyard. So shortly after the station was built, the glacier was sort of right there. I mean, you could look out your window and really step out the back door and be on the glacier. And it just keeps moving back and back and back. Not only is it receding, but it's shrinking in height. It's shrinking in height tremendously. You go out, you listen. It sounds like thunder and a thunderstorm because the glacier is constantly cracking and breaking. And back in the 80s when I first went to Palmer Station, you would go in the backyard and you'd be lucky if you'd see the glacier calve maybe once a month. Now it's like every 20 minutes. The glacier is calving and cracking and yeah, very scary stuff. So the evidence is it's all over the place. The signs of rapid warming are everywhere. This was an ice cave that, again, when I was at Palmer Station, met my husband, Pat Hassett, at Palmer Station back in the 80s. We used to go over to this area where there was this magnificent ice cave. And when I had my children and went back to work in the Antarctic almost a decade and a half later, I said, oh, let's go see the ice cave. Let's go back to that ice. I remember that beautiful ice cave. Oh, Lisa, it's been gone for years. It's been gone. It's melted. It's gone. So this ice cave is no longer. Again, glacial retreat, scouring of the rocks, smoothing, greening of the Antarctic. There are mosses and grasses that are just proliferating over regions of the continent that were never there before. Lots of winds. It's like, the same phenomenon as these hurricanes. With the warming of the water, the ocean temperatures have warmed considerably over the last 50 years. And so it's a much windier place. There's much more precipitation. The same, again, phenomenon that's happening in the Caribbean. And so the question we're addressing in our research is, how will these Antarctic fishes fare in a warming world? Because one of our concerns is that right now, or maybe in the past, the balance between oxygen demand and oxygen supply it's been balanced. What's going on here? Okay, my computer's not behaving, sorry. But as the oceans warm, that balance is going to be tipped. Remember, as we increase temperature, we decrease oxygen solubility. There's not going to be as much oxygen available to these animals as the temperatures warm. So this is a big concern. So I'm sorry, my computer has just completely died on me. Okay, yeah, so with temperature increase, we're going to get a mismatch between our oxygen demand and oxygen supply. And our data suggests that the ice fishes, these animals that lack hemoglobin, are surely going to be the most vulnerable. Okay, I think I don't really, well, do I have time? I want to leave some time for my students to talk. So I'm going to skip over a couple slides, and I'm going to leave them in your hands, or leave you in their hands, to say a couple things about our current research. So what we're asking in our current research is two fundamental questions. First, what are the physiological and biochemical processes that are most sensitive to warming? So is it the cardiovascular system? If it is the cardiovascular system, what specifically in the cardiovascular, what part of the cardiovascular system is limiting their ability to tolerate warming? Is it something to do with the membranes that Amanda will discuss? And then the second major question we're asking are, what are the capacities of these systems, the cardiovascular system in particular, to make adjustments to warming? If we give the animals some time, if the change is not so rapid, will they be able to make adjustments in their metabolism to cope with this warming? So Elizabeth will talk about briefly these cardiovascular, some of the data that we have on the cardiovascular system.
Okay, thank you. So like Lisa said, we're approaching this question from multiple perspectives. And in this case, one of our collaborators, Stuart Eggington at the University of Leeds, looked at heart rate as we warmed the fish. So he surgically inserted an ECG graph into their hearts, and we slowly heated the fish over a constant temperature, whole body warming. And here, as Lisa's showing, so as we increased temperature, we found we could measure their heart rates and their heart rhythms with this monitoring device. So here, as we heat the fish, their heart rates increase, which makes sense. As oxygen availability decreases with increasing temperatures, we would expect that they would have increased heart rates to pump out oxygen at a faster rate to compensate for that. What's really interesting is we found that the ice fish increase their heart rates more quickly, and they also enter into a state of arrhythmia, which means their hearts beat erratically at a lower temperature than that of the control group, the red-blooded fish. So here you can see they're labeled ACE and CORE on the graph. ACE is the ice fish, And here is this dashed line is the ice fish, and their hearts enter into arrhythmia at a much lower temperature than the red-blooded fish. This leads us to believe that there's something going on the cardiac level that's limiting their overall thermotolerance.
As Lisa mentioned, another focal point of our research is biological membranes. Now, membranes are the lipid barriers that surround cells and organelles of cells, and they're really important for regulating the intake and outtake of different molecules. They're also important in coordinating cell signaling, and maintaining the shape and boundaries of the cell. Membranes are made up of phospholipids, and phospholipids, like many other lipids, are very sensitive to temperature. And so we've brought in a small demonstration to show how lipid fluidity is altered by temperature. And so these mason jars that Lisa and Elizabeth are holding have olive oil, and the container that Elizabeth is holding has been kept at room temperature, and Lisa's bottle or jar has been held has been chilled for the last several hours. And I know it's a little bit difficult to see, but the Lisa's jar is is a bit more viscous. It moves less readily than the oil that's been held at room temperature. And like olive oil, membrane lipids also move, and membranes function best within a certain temperature range. If the membrane becomes too cold, like our oil that's been chilled, it can become gel-like. And for the membrane, this means that diffusion is hindered. But if the membrane becomes too warm, if we heat it up too much, it can become disordered and reach a hyperfluidized state. And at this point, we see loss of membrane integrity. And so, like Goldilocks, the membrane can become too cold or too hard or too soft. What we really want is for it to be in this ideal, just right state. And so for ectothermic animals to be able to survive, their membranes have to maintain this ideal fluidity state at the temperature at which they live. These are some data that are part of my graduate work. Part of my work is studying the membrane fluidity of cardiac mitochondria. So mitochondria that was prepared from the muscle of the heart. And I measure membrane fluidity using a technique called fluorescence depolarization. And essentially what this means is we take a fluorescent probe called diphenylhexatriene, or DPH, and we intercalate it within the bilayer. And then we excite the probe at a certain wavelength, and then it emits a value at another wavelength. And this is called its polarization. And the polarization value is a measure of the membrane fluidity. For the purposes of this presentation, I'm referring to these data as the membrane viscosity. And as you can see, in these data we measured membrane fluidity from 2 degrees up to about 30 degrees Celsius for 2 species, Notothenia crioceps, which is our red-blooded notothenoid, and then Kinocephalus oceratus, our ice fish white-blooded species that we've mentioned earlier. And as you can see from these data, as the temperature as the assay temperature increases, membrane viscosity decreases for both species. And so what this means is that as you're heating up the membrane, it becomes more and more fluid. This isn't surprising. It makes sense based on what we know about how these lipids behave. But what's really interesting that we found is that the membrane fluidity of this species, the ice fish species, has significantly lower viscosity, than that of the red-blooded species. In other words, the cardiac mitochondrial membranes of the ice fish are more fluid than the red-blooded cousin. And this is really interesting because, as Lisa alluded to, the ice fish species has a lower upper thermal limit than that of the other red-blooded species. And as I said before, membranes, as they become more and more fluid with warming, the membrane starts to lose functionality, and this can affect the function of the cell. And so as the membrane becomes more and more fluid, it does reach this point where the animal can no longer survive. And because the ice fish membranes overall are more fluid, it's very likely that they might hit that point of loss of functionality at a lower temperature than the red-blooded species. And so these data indicate that membrane fluidity might help to account for differences in thermal tolerance among Antarctic nodothenoids.
Okay, so one last thing that I want to leave you with is something to think about is the final challenge that I'm going to discuss with regard to this wonderful group of animals, and that is the pressure from commercial fishing. So the Antarctic toothfish is it's the largest Antarctic nodothenoid species that exists, and it's large, like up to 200 pounds. I mean, we caught animals that were 150 and so on pounds. Now, this fish is currently being fished as Chilean sea bass. So the Chilean sea bass that was formerly sold in the fish markets all around the country and all around the world for that matter is a close relative. It's actually the Patagonian toothfish. So it's the same genus, a different species. It looks similar. It's smaller, but it's very similar. So this shows data from the UN, from the Food and Agricultural Organization, and it shows the data, the catch for this Patagonian toothfish that went up and up and up and then collapsed in about the early to mid 90s. This was at the time that the arm of the Antarctic Treaty, the Convention for the Conservation on Antarctic Marine Living Resources, decided to open the fishery for the Antarctic toothfish. This fish, if anybody saw the Last Ocean film about a year ago, this is the highlighted species in that documentary. So there's a lot of concern right now that this fish is being commercially over-harvested. A lot of the fishing is illegal. It's estimated that about 70% of the fishing for this species is illegal and unreported. How to police this in the Southern Ocean, which is so far away and so rough and cold and so forth, is a real challenge. If we look at other fisheries for other species, so there were some other species that were fished as early as the late 60s. This one, Nodothenia rossii, was fished off South Georgia, I believe. The fishery collapsed and was closed in the 80s. It's still, to this day, at less than 5% of its original stock size. So this fish is not recovering. Another example is the gunnerii. This is an ice fish species. So this is one of the hemoglobinless fishes. It had a relatively short fishery. Again, it has not recovered, even though the fishery has been closed for almost 30 years. So these are very slow-growing animals because they're living at such cold body temperatures, and not enough is known about their biology and their sustainability. What is a sustainable catch limit for these animals? So I'd like to thank a lot of people who've been involved in the research, including my graduate advisor, Bruce Seidel, who really started this project back in the 80s, and I went south with him to the Antarctic again numerous times. My current collaborator, Kristen O'Brien, who's at the University of Alaska, and an academic sibling, so we were all trained by our graduate mentor, Bruce Seidel. These are my past graduate students and current graduate students, Amanda and Elizabeth. And the National Science Foundation, as I mentioned earlier, supports our research. I got an early start thinking about Antarctica because my father, Freddie Crockett, went to the Antarctic with Byrd back in the 20s, 1928 to 1930. So I grew up hearing stories about the Antarctic, and it was a place I always wanted to visit myself and try to build a career around. So I'm happy to take questions and come up and see if you didn't get a chance to see any of the inverts. Yeah, come on up. Yes.
I have a question about something that you did in the lecture, the traumatic So I understand why above ground, sort of standing out would be a trait with negative survival value. You're a predator, your prey can see you stalking them. You're a prey animal, you know, a polar bear can see you from a mile away.
Right,
Why
right.
isn't that the case
Under the ocean. Yeah, that's a great question. So one of the reasons that that's not a problem under the ocean.
Question?
Oh.
Because you're getting taped.
Okay, so the question was, it seems obvious why the animals above the surface are more monochromatic, black and white and so forth, because that's cryptic, they're not visible. But then why wouldn't the animals that are living on the seafloor have similar, more monochromatic colors? And one of the reasons for that is that not all wavelengths of light survive at the deep depth. So for example, red appears black at the depth where many of these animals are found. So they appear colorful to us, but in nature, they're not visible. So that's a great question.
Robot on your shirt.
A little unrelated to the presentation, I'm curious, what are the invertebrates preserved in there?
Yeah. So what are the invertebrates preserved in the jars? So we have, this is a polychaete. So this is in the family, or the phylum Annelida. So this is an annelid. This is a polychaete worm. These are brachiopods, so they're like a bivalve, like a mollusk, but they're actually unrelated. So they have two shells, like a mollusk, like a clam, but they're not related to clams at all. So they're brachiopods. Do we have some others that are floating around? Spiders. So this is a sea spider, and a sea spider is in the phylum Arthropoda. It's related to, although distantly, horseshoe crabs. It's related to crabs. It's related to shrimp. It's related to, who else is in Arthropoda? Insects, spiders. Yeah, great question. And if you want to come take a look. So they're all labeled. So.
We know that the critical thermal maximum of these animals, there's some slight variation between species. For the ice fish, it's about 13 degrees Celsius, and for the red-blooded species, it's about 17 degrees Celsius. Now, that being said, whether the membrane becoming completely non-functional is the reason that animal has reached its critical thermal maximum is difficult for us to say. In that data that I showed you, one thing that we found that was really striking was that we didn't really see this point where the membrane goes from being really solid to going to really, really fluid really quickly. And we're not sure exactly why that is. Sometimes the membrane goes through that abrupt transition and sometimes it doesn't. So it's difficult for us to say for sure.
That's a great question. So the question was, are there any fish species that we should avoid buying? Yes, you should avoid buying Chilean sea bass. So Safeway has stopped selling Chilean sea bass. Kroger's has not. Kroger's doesn't have Chilean sea bass every day, but they have it now and then. So for sure, avoid buying Chilean sea bass if you can. It's delicious, but that's part of the problem. People have a huge appetite for it. Yeah.
This is regarding the future of global climate change. Will the melting of the ice lower the amount of salt in the water and raise the freezing temperature?
That's a great question. So the question was, will the melting of the ice change the freezing point because it's going to make the water more fresh? Yes, the answer, yes, it will. Yes. Yes, and it could also have an impact on the fish as well, because these fish are adapted to full-strength seawater, and a more dilute seawater could potentially be very problematic. Great question.
Kind of going with the whole like temperature questions. Obviously, you guys have found like thresholds of where the fish is kind of like their temperature limit. Do you have like a good estimate of like what the ocean temperature, like what's the cutoff and like the point of no return for these fish? And like, if so, how long do you think until we reach that?
Great. Okay, so the question was, can we predict?
We can't hear you.
Can we predict what temperature these animals can survive to, and what's sort of... So the answer to that is we're working on it, I think. So it's going to depend on the species. So the ice fish may be less resilient to warming than some of the red-blooded species. Our data would suggest that, but we're still really trying to tease that apart. One of the things that Amanda was working on this season at Palmer Station was an acclimation where we actually brought the animals into the lab. We kept some at their normal temperature, and then we kept half of them at a much warmer temperature. And now we're just analyzing some of the data, or analyzing some of the specimens, the samples that we brought back, to determine whether they can really survive and make the adjustments they need to for that. So that's a great question. Yeah, you're welcome. Yes. Wait, this. Yes.
haven't talked much about birds. I was wondering how the snowbird, and I saw another bird flying by in one of the fishery pictures,
Yeah.
how their wings aren't frozen, how they're still functional at that temperature.
How their wings aren't frozen because they're coming in contact with seawater. So their bodies are warm. I mean, it's a great question. Their bodies are warm. They're what we call endothermic animals. So they're circulating warm blood to the periphery, although they don't circulate as much blood to the periphery because they don't want to lose a lot of heat through their appendages and their extremities. So their extremities are actually colder, but they're not so cold that they're below zero. They're still above zero, so they're not going to freeze. And even the penguins, what they do, when they're standing on the ice, they stand on the ice and they raise their feet up so that their feet aren't as much contact with the ice. So that reduces the thermal exchange and keeps them from losing too much heat to the ice.
So Lisa, I'm wondering about the challenges to our wonderful researchers. When you go diving and get these samples, how long do you do? You do it?
No, we don't dive. No, we don't dive for the samples. We collect the samples mostly from the research vessel, although Amanda did a lot of fishing by hook and line off the pier. But we're not. There are other researchers that use diving, but that's not something we're. Yes. Hi, Liam. Well, they're, for the most part, unrelated, with the exception of the eel pouts. There are eel pouts in the Arctic. So the question was, how are their traits different from species in the north, in the Arctic? So most of them are unrelated. There are antifreeze proteins in Arctic fishes as well. And what's interesting is that there's an example of biochemical convergence. So some of the proteins are the very same proteins, but they've evolved independently to prevent the animals from freezing. In terms of activity patterns, we see some of the same activity patterns in Arctic fishes. Some of the Arctic fishes are very active, even at cold temperature. As far as lack of hemoglobin, there's no comparison. So those ice fishes are unique on the planet as being the only vertebrates that as adults lack hemoglobin. So that's a unique feature of Antarctic fishes.
So if we could,
question.
let's thank Lisa one more time.